Hello, everyone. I'd like to welcome you to our next session today, which is the first of two sessions about pathogenesis and immunology. This one will be hosted by myself and Dr. Margarita Says. And when asked to simplify, I tend to say that studying disease pathogenesis is basically figuring out what happens when and where in an affected animal. And if you then have time and money left over, you can start working on the hows and potentially what you could do to interfere or prevent. And although that's clearly oversimplifying things, I think it's very relevant to recognize the importance of what is often considered very basic pathogenesis studies. And those are usually the studies done involving animals. Because in order to fine-tune our FMD countermeasures and to make appropriate use of the advanced molecular technologies that we now have at hand, it's still important to understand the basic biology of disease, including such seemingly simple details such as knowing how animals get infected and where the virus is replicating at what phase of disease. Also, one of my personal pet peeves is the difference in FMD pathogenesis between different host species. And specifically, as we heard, pigs are not little cows, but even within ruminant species, there are actually differences in the tissue localization of virus, both in early and late stages of infection. And to tie into some of the discussions that were going on the sideline in the chat yesterday, and that were also touched upon in Dr. Art's presentation just now, knowing the site of initial infection for the host species that you're working on, and that oropharynx and nasopharynx are actually not the same thing, it's critical, for example, for designing challenge models that appropriately will evaluate your vaccine and your biotherapeutical. Of course, these days, the concept of studying pathogenesis have broadened as we are now able to investigate specific host virus molecular interactions using in vitro systems. And in this session today, we are taking a step in that direction as we'll be looking more into the molecular aspects of FMD infection and host response. Our first invited speaker today is Dr. Zidong Zhang, who's a professor of veterinary virology at the College of Animal Husbandry and Veterinary Medicine at the Southwest Minzhou University in China. Dr. Zhang received his PhD degree from the Royal School of Veterinary Studies in Edinburgh. And since then, his main interests have been studying the pathogenesis of viral diseases of livestock, with emphasis of foot and mouth disease virus, peste petite ruminants, and pox viruses of sheep. He's in the past worked as a group leader in the vesicular disease pathogenesis group at the Pirbright Institute, as well as a section head of vesicular diseases in the National Centers of Foreign Animal Disease in Canada, and as a program head of viral disease in grazing animals at the Lanzhou Veterinary Research Institute in China. And for all of us that have worked in the field of FMD for some time, we are very well aware of the massive contributions of the work of Dr. Zhang to our general knowledge of FMD pathogenesis and host response. And in this talk today, Zidong will talk about how stimulator of interferon genes is required for foot and mouth disease virus-induced autophagy. Thank you.
Hello, everyone. And first of all, I would like to thank GFAA meeting organizers to give me such good opportunities. In today's presentation, I'm going to present some data about the role of stimulator of interferon genes in induction of autophagy during the FMD virus infection. STING is an important immunoregulated protein which is located in the cells, and it's best known for its very important role in the host defense against DNA virus infection by inducing interference production. And also some studies also show that deficiency of STING in the cells and the mice greatly facilitated replication of several RN viruses, such as WSV, dengue virus, and West Nile virus. So it's apparently now the STING is also played a very important role in the host response against infection with iron virus. But however, how the sting is engaged in the antiviral response to the infection with iron virus is not well documented. And our studies show that sting is engaged during FMD infection. And we have demonstrated that sting is essential for autophagy activation during the FMD infection. Here you can see that when we constructed sting knockdown cells and then infected with the FMD, and it didn't see the autophagy can occur. And at the same time, virus replication dropped. So we want to know how the sting activation induces autophagy during FMD infection. And to test whether sting activation is necessary for the induction of the autophagy during FMD infection. And first we'll test it. Part of the activation steps for the sting is sting trafficking. And we tested, which first treated the cells with two different protein trafficking inhibitor. One is CGA, another one is BFA. And from here you can see that when FMD infected cells treated with these two, either GCA inhibitor or BFA inhibitor, and FMD infection still can induce autophagies in these cells when compared with cells without treatment. Here you can see the autophagies here, and also you can see the autophagy occurred in the cells treated with the protein trafficking inhibitor, GCA and BFA as well. But this result also further confirmed by co-staining of sting and ERGIC with protein marker 52. Here you can see this red color is ERGIC 53, specific marker for ER-Golgi intermediate compartment. And here's the sting in green, and when you look at the overlap image, and you can see there's no trafficking of sting from ER to Golgi ERGIC compartment. So this result suggests that sting trafficking is not required for induction of the autophagy during FMD infection. And activation of the sting also required palmitoylation and Golgi. And so we are using a very specific inhibitor, H151, which was reported to be able to block sting palmitoylation and by covalently binding to the sting and the transmembrane settling residue and position 91. But however, this inhibitor only works on the human and the mouse cells. So to make porcine cells PK15 response to this inhibitor, we are first of all constituted human sting in the sting knockout out PK15 cells. So here, I showed here, when FMD infected cells which expression human sting and still can induce activation of the autophagies here, and this negative without this uninfected cells is infected cells, which means human sting is working in the porcine cells. So after that, we treated these cells with H151 and to look at what happened to the autophagies. And here you can see this control infection without treatment with the inhibitor, and these cells are treated with the inhibitors. You can see the FMD can induce autophagies occurrence in both cells. So which means STING activation by the polymethylation is not required for induction of autophagy during FMD infection. And also STING activates TBK1 with its C-terminals domain, and the truncation of the mutants of the STING1 340 couldn't activate TBK1. So we constructed STING 142 mutants, and then expressed in the poster cells. After that, we're infected with FMD, and here you can see FMD infection still can induce autophagy occurrence in the cells expression STING 1 to 340 mutants. So which means activation, trafficking are not necessary for induction of the autophagy during the FMD infections. And in addition to the trafficking and polytoylation, and STING activation also required polymerization. And activated STING polymerization is stabilized by disulfide bridge and its C148. Mutation of C148 to alanine will disrupt STING polymerization. So we are hypothesis that STING polymerization participates in regulating activation of the autophagy. So to test it that, we reconstituted STING C148 mutants in the STING knockout PK15 cells. And here we successfully engineering cells expressing STING C148 mutants. And then we infected these cells with FMD. And from here, you can see that this uninfected cells and this infected cells. So you can see there's no difference because there's no autophagy observed in this two cells group. And this result was further confirmed by a negative page result, which showed here that the FMD infection can lead to STING polymerization. So this result, this result indicates that STING polymerization is necessary to induce autophagy activation. And our previous study also found FMD infection leads to reticulophage. And here you can see that when this markers is a specific receptor for the reticulophage, and if you knock down this receptor, and then after that infected with FMD, there's no autophagy occurred, and also virus replication dropped. And in the past, well, also our previous study also demonstrated that FMD VP2 protein is responsible for induction of the autophagies during the virus infection. So to test it with the VP2 is also responsible for STING-dependent reticulophage induction. So we knock down, we'll knock down this specific marker for reticulophage, and then transfect it with the FMD VP2. From here, you can see that in these cells, these knockdown cells, and you didn't see autophagy occurred, and which is exactly the same as uninfected, transfected cells. Similarly, and when you transfected STING knockout cells with the VP2, and also you didn't see autophagy can occur. even you see the bands of the VP2. So which means indicate VP2 is responsible for STING dependent reticulophage induction. And because when the virus get infected cells, and the cells will sense the viral RNA in the cells. So we test whether the viral RNA is good enough to induce STING-mediated autophagy. So we'll do the two things. One is we'll transfect the cells with the viral RNA, which no virus protein will be translated. So when you get this, there's no protein, virus protein can be produced. And here you can see this transfected cells and this control that you didn't see autophagy occurred. And when you transfected the cells with viral RNA, which viral protein L, VP4, VP3, and VP2 can be translated, and then you will see the autophagy can occur. So which means FMD virus itself is not sufficient to trigger STING-mediated autophagies. So the next question is, how about, because when the virus RNA get into the cells and the RIG-1, which is RNA, foreign viral RNA sensor, will sense the viral RNA derived from RNA virus. And so here you can see that even we knock down the RIG-1, and then you didn't see the autophagy occurred. And then when you put back the RIG-1 and then overexpress them, and then you can see that the autophagy can occur. But when you use RIG-1 mutants, which cannot bind to viral double-stranded RNA, and then you still can see the autophagy can occur. So which means virus binding to RIG-1 is not necessary for FMD induction autophagies. And in conclusion, STING trafficking activation is not necessary for autophagy activation, and STING polymerization is required to induce autophagies. FMD virus RNA itself is not sufficient to trigger STING-mediated autophagies. Finally, I would like to thank all the members from Southwest Minzu University and Lanzhou Veterinary Research Institute. And I also would like to thank the funding organization giving money to do this work. And thank you very much, and thank you.
Okay. Hello, everybody. My name is Margarita Sari, and it's a pleasure for me to act as co-chair of this, with Carolina, of this pathogenesis and immunology session. And now we're going to listen to the three selected oral presentations addressing such relevant issues in the field as tropism determinants, viral RNA infectivity, or the molecular basis of persistence. So I'm going to introduce our three speakers and the titles of their corresponding talks. Our first speaker will be Soumendu Chakrabarty, talking about foot-and-mouth disease virus non-structural protein 3A modulates cellular innate immune responses to influence host tropism. Next speaker will be Michael X. Baumer with a talk entitled Infectivity of Full-Length Genomic RNA of FMDB in Cattle. And our last speaker will be James Su with the talk Mechanisms of Establishment of FMDB Persistent Infection Inferred from Differential Tissue Gene Expression during transitional phase from acute to persistent infection. After the their talks, we'll have the chance to start a discussion session, which we hope will be very interesting and productive. Okay, thank you, everybody.
Hello everyone. It gives me immense pleasure to welcome you all to my presentation on FMD3 non-structural protein, how it modulates cellular antibody response to influence host tropism. We were working on the 3A non-structural protein for the last few years and were interested how a deletion in this 3A protein influences host tropism. Coming to the picornavirus 3A non-structural protein, it is highly divergent across all the picornavirus genera. It is important in formation of replication complex. It also enables cellular functions to promote virus replication. But we are more interested, especially on how the 3A protein influences host tropism. Previously, it has been seen that these mutations within this 3A protein has been associated with host tropism. Coming to FMD3A protein, it is having 153 amino acid and is the biggest among all the viruses within this picornavirus family. The N terminal is highly conserved while the C terminal is variable among the 7 serotypes. Partial deletions in this C terminal region of the 3A protein has been associated with disease in pigs. There has been several publications associated with these 3A deletions and pig-specific outbreaks. There has been natural deletions that has been observed in the outbreaks in the field. Recombinant viruses have also been generated having various length of this deletion within this C terminal of the 3A protein, and experimental infections have also shown that this 3A deleted virus does not produce a clinical disease in cattle. In our lab, we are having a naturally occurring 3A deleted virus from 81 to 137 amino acid deletion in this 3A protein. The virus belongs to the serotype O, Manisha subtype. We were also having the full-length 3A version of this virus. We are interested in characterizing our virus in a pig and a cattle cell line. The pig cell line IBRS2 is having a non-functional interferon signaling pathway, where the bovine cells were a primary bovine thyroid cells expected to have a fully functional interferon signaling pathway. We did a competition passage in these two cell lines, initially taking equal proportions of these two viruses. In IBRS2, after few passages, the 3A deleted virus had a competitive advantage over the full-length 3A virus. After passage level 5 in IBRS2 cells, only the 3A deleted virus was observed. In BTY cells, after passage level 2 only, the full-length 3A virus was seen and the 3A deleted virus was not observed. We then did an infectivity assay in the IBRS2 and the BTY cells. The titer of the 3A deleted virus was higher in IBRS2 cells compared with the full-length virus. In the BTY cells, there was a huge difference between the titer of the full-length virus and the 3A deleted virus. The titer of the 3A deleted virus was slightly more than the input indicating that the 3A deleted virus is attenuated in bovine cells, which is having the functional interferon signaling pathway. Now this raises a question whether the cellular antibody response plays a role in this attenuated phenotype in the bovine cells. This is an overview of a typical interferon signaling pathway in a cellular environment. The PAMP or pathogen-associated molecular patterns consisting of a single-stranded or double-stranded viral RNA is sensed by cytoplasmic sting or mitochondrial RIG-I or MDF5 signaling pathway to activate the production of type 1 and type 3 interferon. The interferon produced acts in an autocrine or paracrine manner in the neighboring cells through the ZAC-STAT kinase pathway to release hundreds of interferon-stimulated genes or ISGs. Some of these ISGs have antiviral functions. If you are using a drug, ruxolitinib, or Rux simply, it blocks the ZAC-STAT kinase signaling pathway to stop this production of the ISGs. So we repeated the TCID50 experiment in the IBRS2 and the BTY cells in the presence of Rux. In the IBRS2 cells, there was no significant improvement in the titer of the 3A deleted virus. But in the BTY cells, there was a significant improvement in the titer of the 3 deleted virus in the presence of RUX. There was at least a log improvement in the titer of the 3 deleted virus in the presence of RUX in the BTY cells. So we then performed a cell killing curve of the 3 deleted virus and the full-length 3 virus in the BTY cells. The progression of CP was observed by the reduction in the cell mononuclear confluence over time in a live cell imager, the IncuSite. The full-length virus achieved complete CP by the end of the experiment, whereas the 3 deleted virus hardly reached full CP. But on addition of RUX, the 3 deleted virus also progressed to complete CP by the end of the experiment. Coming to the summary of the present study, we have demonstrated that the FMD with a deletion in the C-terminal region of the 3A protein have a competitive advantage in the porcine cells, which suggests that this may be due to its enhanced replication. The 3A deleted virus is attenuated in the primary bovine cells having a functional interference signaling pathway. The attenuated phenotype on the 3A deleted virus can be partially reversed in the presence of interference signaling inhibitor drug RUX. The findings raises a question that whether this species-specific phenotype of this 3A deleted virus in bovine cells is mediated by interferon-stimulated genes. I'd like to acknowledge all the members of the piconavirus molecular biology group, Stephen, Chris, Jamie, Vicky, and Audrey, and the past members Joe, and also the colleagues from the Purbat Institute who has helped in this work. Thanks all. I'm ready for taking questions. Thanks. Thank you all.
Hi, I'm Michael Eschbaumer from the FLI in Germany, and I would like to present data from a recent animal experiment in which we assessed the infectivity of full-length FMDV RNA in cattle. Now, why did we do this experiment? I think you're all familiar with lateral flow devices that are used for the pen site detection of FMDV antigen. It has been convincingly shown by the EU Reference Lab for FMD that the virus and positive LFDs can be reliably inactivated by immersion of the LFD in a low pH citric acid solution. After the treatment, no infectious virus remains on the LFD, but the FMDV RNA is still present and can be used for confirmatory PCR in a laboratory, sequencing, or even virus rescue by transfection of cell cultures. Based on the safety data, UFMD has recently issued an opinion confirming that LFDs treated with citric acid are not dangerous goods under IATA rules, which is very handy for people who want to ship them from an endemic region to a reference laboratory, for example. I was part of the group drafting that opinion, and I do agree with its conclusions, but it is important to keep in mind that many things can go wrong with packages in the mail in all parts of the world. And in the worst case, these inactivated LFDs can come in contact with animals that are susceptible to FMD. We know that on one hand, there are absolutely enormous amounts of FMDV RNA on these devices. And on the other hand, only one intact FMDV genome needs to get into a cell to start an infection. So these are pretty good odds. In the recent literature, there are a number of studies where FMDV RNA has been injected into animals, but in all cases that I could find, transfection reagents such as lipofectin were used to make sure that the injected RNA actually got into cells. So that was not useful to answer our question. There's a study on rabbit hemorrhagic disease virus as well, another positive strand RNA virus, but they also used transfection reagent. The only study that didn't was for PRRS virus in pigs, and that one was successful too. So we decided to do our own study. We compared different virus isolates for their transfectability, for lack of a better word, and ultimately settled on AIRAC22, one of the reference strains that we use in our serum neutralization tests. We isolated high-quality RNA using trizole and isopropanol precipitation. The fresh RNA was then injected superficially into 4 sites on the tongue of 6 deeply sedated steers by the standard method. Of course, we made sure that the preparation contained a lot of FMDV RNA, but no infectious virus. Keep in mind that what is being quantified by the real-time PCR here is not all full-length genome, of course. When we inoculated the RNA preparation into cell culture without a transfection reagent, there was no effect, but infectious virus was efficiently recovered when we did use lipofectomy. On the day after the inoculation, we could not sedate the steers again due to our animal protocol, but we did take their temperatures and we tried to look into their mouths. And it's hard to say with certainty, but I do think that I saw vesicles on the tongues of some of the animals on that day. And we also noted that some of the animals seemed to be salivating more than usual. All animals had elevated temperatures on day 1, which further increased on day 2. And based on our clinical score, which includes fever, lameness, feed intake, and attitude, one animal had to be euthanized on day 2. The others were sedated on day 2, and we did full clinical exams, revealing large primary vesicles on all the tongues and secondary lesions on all the feet of all animals. The clinical picture was quite stunning, actually, particularly if you keep in mind that this was not a recent field isolate or cattle-adapted virus, but an old laboratory strain that has been passaged in tissue culture many, many times. We also took saliva and serum from these animals, wherein you can see a clear increase in the FMDV RNA content over time. Infectious virus was also re-isolated from all saliva and all serum samples on days 1 and 2 from all animals. And the vesicular material collected at necropsy was also highly positive in the FMDV real-time PCR. So in summary, purified FMDV RNA is highly infectious for cattle, and any exposure of susceptible animals to such RNA should be avoided. However, I know that injection is a highly artificial way of exposure, and we would like to try a more realistic route next in order to collect more data for a precise assessment of the risks involved. So I'm interested to hear your opinion on what route would be most appropriate. Going back to the EU-FMD joint opinion, it is made very clear that when shipping inactivated LFDs, this has to be done in a way that allows you to track where your shipment is and make sure that it's not lost in transit. That's it for now. Thank you for your time, and I'm looking forward to the discussion.
Thanks for accepting this presentation. After FMD infection, 50% of ruminants become persistently infected in pharyngeal epithelia. But the persistent infection is not found in pigs. The immune mechanism and causes are unknown. We use gene expression profile to understand the mechanisms. The study has been reported in 4 publications. The objectives of this study are to infer mechanisms involved in the establishment of persistent infection and to hypothesize the causes based on the data collected during the transitional phase. Down-regulated expression of a group chemokines called ELR plus CXCLs was found to be associated with FMD persistent infection in our study. The function of these chemokines is to recruit immune cells to clear pathogen in infected tissue. Among these chemokines, CXCL15 was differentially expressed between carrier and not carrier in the micro-dissected epithelia. Water buffaloes were more susceptible to FMD persistent infection than cattle in a study in India. Interestingly, CXCL15 gene is not functional in water buffalo due to an early start codon. The expression of ELR plus CXCL chemokines is known to be induced by IL-17 produced by Th17 cells. HR signaling inhibits Th17 response in mucosa. through inducing Th17 to Treg transition to reduce IL-17 production and inhibiting canonical NF-κB signaling pathway to suppress IL-17 signaling. Three major immune mechanisms was found to be associated with persistent infection in this study. First, carrier-initiated Th17 response and had more Th17 cells in epithelia than terminate during the transitional phase. Second, the level of Th17 cytokine was not increased in the carrier as expected, and the downstream genes ELR plus CXCL chemokine was expressed at lower level in carriers, indicating that Th17 activity and IL-17 signaling was suppressed in carrier. Third, the activity of AHR signaling in carriers increased as compared to terminates, which suppressed Th17 response. Two Th17 stimulating cytokines and two Th17 recruiting chemokines and a receptor suggest that carrier initiate Th17 response and add more Th17 cells in epithelials of the carrier than terminators. However, IL-17 cytokine level was not increased in the carrier, indicating that Th17 cells was suppressed. AHR expression was significantly upregulated and HIF-1A was downregulated in carriers, suggesting higher AHR signaling activity in carriers than in terminators. Several differentially expressed target genes of AHR and HIF-1A supported increased AHR signaling in carriers. 6 Treg-associated genes upregulated in carrier but not upregulated FOXP3 suggested AHR-mediated Th17 to Treg transition. The differential expression of genes in the NF-κB signaling pathways supported AHR-mediated immune inhibition of IL-17 signaling in carriers. In summary, our results indicate that transactional carrier initiated Th17 response, but the response was suppressed by increased AHR signaling, which reduced the expression of chemokines needed to recruit immune cells to clear FMD and allow the virus to persist. Therefore, the question is what possible ligands could activate AHR in carriers? AHR ligands are signobiotic chemicals containing aromatic hydrocarbons. There are two sources of AHR ligands, dietary and cellular. Dietary AHR ligands are derived from some plants, bacterial tryptophan metabolites, and dioxin-like pollutants, which are closely related to luminance, as exemplified by their plain diets, pathogenesis called fog fever caused by excessive production of AHR ligand in the lumen, and the highest level of dioxin-like toxin found in beef among meats and poultry. Luminant major energy sources, short-chain fatty acid, has been reported to increase the responsiveness to AHR ligand stimulation through increasing AHR expression. Therefore, based on our result and the luminant physiology, it is hypothesized that high short-chain fatty acid production in lumens increase the sensitivity of luminance to AHR ligand stimulation, and excessive luminal AHR ligand production cause FMD persistent infection. This hypothesis could explain why luminance but not pigs are susceptible to FMD persistent infections, and why vaccine cannot cure FMD persistent infection. This research was supported by U.S. Departments of Homeland Security and Agriculture. Thank you for your attention.
Okay, thank you very much to our four speakers. And now that's fine with you, Carolina. Let's go to the questions. Okay. So let's start with the first question for Sidon. Thanks for your talk. What viral components are triggering autophagy? Does it occur through a different mechanism apart from through RLR's recognition? In which cells you tested autophagy?
Okay, thank you, and it's a good question. Yes, and in our hands, our experiment demonstrated that FMD-VP2 is the key element or component to activate autophagy. So, which means, of course, because it's a protein, we have some data to show that FMD infection, which can cause the ER stress to activate autophagy. So I think that this is probably slightly different because on your hand, RNA itself is not enough to activate autophagy. So it's for sure it's different single pathway to activate autophagy by through the FMD protein. So it's we think it's mainly FMD infection causes ER stress and induced autophagy. And we use the PK pulsar PK50 cells line. Hopefully, I answer your questions.
We have another question for Somendu from Michael asking about if you tried the experiments in primary porcine cells.
We've not tried in primary porcine cells, but we have used PK15, which is having the functional interferon pathway. So we have used and we have done the cell killing curve assay in the PK15 cells also. And we have seen this particular phenotype still remain in the porcine cells. So this is specific phenotype. So independent of the cells which you are using, in the cells which we have used for the porcine is the Iberis2 as well as the PK15. And we have seen the same phenotype of this 3A related virus in the cells. I hope this answers your question, Michael.
Okay,
Yes,
thank
it
you.
does. Thank you.
Okay, our next question is for Michael, from Michael Bouquet. You did this work in cattle. Do you have plans to try it in swine, perhaps by coating a chew rope with RNA to see the risk to a pig of chewing on a viral RNA containing test?
I like this idea. We had not so far considered doing it in pigs, but as I said in the talk, I'm really looking for practical ideas of some more natural way of exposing the pigs. And but the idea with the chew rope is really nice. I mean, you would need a large volume of RNA to actually have a meaningful amount on each rope, but I think it could be done, and that would be a fairly realistic way of addressing the question. I like it.
I can imagine that we did some recent experiments with feeding infectious FNDV to pigs, and it needed a quite impressive amount of virus to actually make them sick. But I think that the actual time that they are chewing, that they're exposed to the virus, seem to matter. So if you can find a rope that they like to chew on and keep them chewing on it, you might increase your odds.
So mix the RNA with apple juice or something.
And something like that, yeah. Have to optimize a little bit.
Okay, in connection with that question, we have 2 more, so I make them in a row. Hi, Michael, thanks again. When considering the EUFMD position paper, isn't it of greater importance to consider the possibility that there might be a failure in the inactivation process in the field due to human errors? And this risk constitutes the greatest risk associated with the transport of positive LFDs, he asks.
Yes, I absolutely agree. That is a far bigger and far more realistic scenario than the package ending up in some pig's mouth somewhere along the way. And this is actually addressed in the opinion as well, that one of the things that we ask for is that there's always a pH indicator included with like these inactivation kits, so that you can make sure that the citric acid solution that you use for the inactivation has a reasonably low pH, with this being really the critical parameter for the inactivation. And then that the inactivation is only done by personnel that has been trained and knows what to look for when carrying out the procedure. And also they are asked to include some written confirmation that they have actually done all the steps required and that they vouch for the proper inactivation of the LFDs that are being shipped. I completely agree that is the most important point. Yes.
Okay, we have a question for James from Michael. Could this hypothesis be tested by externally activating the pathways in swine that you identified in cattle?
I think it's possible, but at this moment, because I do not put into a lot of thinking in this way, what I need is just try to say what is the possible cause. But because the major difference in physiology between ruminant and pig is because of the lumen. And then the lumen is the one that it's major factor is make the ruminant more susceptible to AHR-lichen signaling. Another one is also produce a lot of the lichen. And the lichen, the amount of lichen production, it depends on the diet and also the bacteria in the lumen, because of 2 factors. And certain bacteria like lactobacillus, because they have a certain enzyme, can metabolize the tryptophan to convert it into several products, indoor products. And those are very strong AHR lichen. And it's been known that AHR lichen can regulate mucosal immunity. There's several publications. And this is part of it's called the microbial and immune system interaction in human, mostly in human or in mammal, except ruminant. Mostly it happens in colon because there's a lot of bacteria and most of the fiber will be digested in a major intestine. But ruminant, it just have the ruminant with their physiology that associate with they convert almost all the fiber and also sugar or anything into short-chain fatty acid. And there are three major short-chain fatty acids: is acetate, propionate, and then butyrate. And these have a different activity. Actually, they bind to different receptors, and that is how they regulate. It's called anti-acetyl. It's a histone acetylase activity, and that's how they regulate the AHR expression. They pump up and different fatty acid. They have different activity because they bind to different receptors. But to answer your question, at this moment, I don't know how to say it, yes, I think it's possible. We can just inject some AHR ligand to the pig, and then, but you need to know that different hosts have different sensitivity to different ligand. Because if you know that human respond to certain ligand strongly, but mouse do not respond, because the evolution and then the receptor, the sequence may change because they need to deal with the certain environment they in order to survive. And right now that is a very hot area. It's the how human currently we live in a cleaner environment, and then we have a mucosal immunity overreact to the pathogens. Mostly they are harmless, but we overreact to it. And this is a part of the microbial immune system interaction.
Okay, there's another one for you. From Rebecca Agarwal. Are you suggesting that dietary modification in ruminants could prevent carrier state?
Yes, it's possible. One is that first one, we know that certain bacteria have that enzyme. They convert tryptophan. But we don't know except lactobacillus have that enzyme. But I think we can use the RNA-seq to do the entire microbiome in the lumen. The other thing is that we can also manipulate the diet because in some are mostly grass-fed and some maybe the corn-based. They can produce different combination of the three major short-chain fatty acids. But at this moment, I think mostly we need to find the sources of AHR. So are they come from toxin or they come from plant by itself, or it's because of the bacteria, they convert tryptophan to that. So that is, it's an open field, yes.
So another question for Michael from Charles Nafon. Can you speculate on how the FMDV RNA gets into the cells in vivo in the absence of any transfection reagents?
Yes, it is only speculation, but I think it's mechanical. Probably it's you're dragging a needle through the epithelium, and I think you're just gonna hit that sweet spot where you just barely nick a cell membrane, not enough to actually kill the cell, but enough to get at least one FMDV genome into the cell. And that's really all you need. You need one intact genome in one surviving cell, and it takes it from there.
Okay, one more for you, Michael, from Luis Rodriguez. Would it make sense to feed pigs with the LFD membranes containing the RNA? This will test the risk of pigs getting infected with LFDs improperly disposed.
Yeah, I mean, this is definitely another option of doing this. It has the advantage of being more relevant to the LFD scenario, but I am afraid, and also going from what Carolina has said, that it has the disadvantage that the pigs are not going to be very interested in chewing on PVC membranes. So that maybe, I don't know, shredding them and mixing them with dog food or something would be the way to go if you wanted to give them the membranes. But yes, I mean the membrane is where it's at. The cotton pads in the LFD and the membrane that has the positive FMDV antigen band. That's where the infectivity is going to be.
So another question that you might already had answered is what transfection reagents you used and what was the RNA concentration?
Mm-hmm. We did not use any transfection reagent. That was the one big difference that we wanted to keep from the other studies that are out there that did use lipofectin or something. We really just used the RNA dissolved in nuclease-free water from the RNA pellet after the precipitation. As for the RNA concentration, since this was not RNA that came from an in vitro transcription, it was total RNA extracted from FMDV infected cells. We did not bother measuring the mass concentration of the RNA, because most of what you're going to measure in like a spectrophotometer or something is going to be cellular RNA. So I do not know the concentration. I just know we took the RNA that we extracted, we had to dilute it 1 in 5 in water to have enough for the 6 cows, and then in it went. A lot of RNA, but I don't have a number.
Okay. Now a question for Soumendu from Sidon. Have you tried using PK15 as IVRS to has a deficiency in interferon signaling pathway?
Yeah, we have used the PK15 also in the cell killing curve, and we have seen the similar phenotype of this 3A deleted virus in this PK15 also, similar to IBRS2. So we have done in PK15 also, the cell killing curve. And the bovine cell line, we have used MDBK also, and we have seen a similar phenotype in the MDB case also, similar to BT-wise. So MDB case is also having a functional interferon signaling system. So in MDB case also, we have seen the similar phenotype of this 3A deleted virus. We have tried two cell lines each, and we have seen a similar phenotype of this virus.
We have another question for James from Teresa de los Santos. Are you planning on doing similar analyses in other ruminants, such as sheep, to prove the hypothesis?
No, at this moment. I think this project, I think, as you know it, Dr. R leading this project. And I think mostly whatever the downstream research will be Jonathan's decision.
Okay, another one for Michael from Donald King. Do you see a role for naked RNA as an alternative to various inoculation for vaccine challenge studies, particularly where cattle adapted virus for a challenge strain is not available?
I mean, that's possible for sure. I'm not sure if there is a clear advantage to doing that, though. I mean, yes, we saw really good, well, not good, fulminant clinical disease, but I'm not sure if we wouldn't have seen the same if we just had used the virus. I mean, yes, the assumption is always it's been passaged in tissue culture a lot of times and is attenuated, is not as virulent in cattle anymore. But if you inject a lot of it into the tongue, I wouldn't be surprised if they got sick from the virus as well. And going back to what we said earlier and yesterday, I think the future of vaccine challenge studies should not be injecting anything, but moving towards more natural routes of exposure as far as they can be standardized.
I think that is all the questions we had. There's a couple of comments in the chat too. Are there any more questions for the speakers? Or is that maybe what we had for this session? This is a moment of awkward silence that was referenced earlier.
Okay.
Everybody's waiting for someone to say something, but I don't think we have any more questions in the chat. So that might be what we had for the session. So maybe we should thank all the speakers for your contributions.
Thank you all for your attention and your talks and your questions. It was a pleasure.
Yes.
Bye.
Thank you.